Okay, so now that we are done talking about antigen, let's now talk about antibodies. So, what are antibodies? So, antibodies are just usually very specific. It only binds to with a specific antigen determinant that stimulated their production. Antibodies are proteins produced by lymphocytes in response to the presence of an antigen. An antibody is an immunoglobulin having particular specificity for an antigen. An antibody is a protein component of the immune system. It circulates the blood, recognizes foreign substances like bacteria and viruses, and it neutralizes them. After exposure to a foreign substance, the antigen called the antigen called the antigen these antibodies continue to circulate in the blood, providing protection against future exposures to that antigen. So immunoglobulins are globular glycoproteins in the blood that participate in the immune reaction. So here is a video about antibodies.
When foreign substances like viruses invade the body, antibodies are triggered to fight infection. Antibodies can also provide some immunity to future infections from that organism. Sometimes, antibodies are donated from people who have recovered from an illness to treat others with active illness. It's been done with diseases including COVID-19. Success varies. Scientists can also create antibodies that target specific invaders. These man-made antibodies can be used alone or with other therapies to help treat diseases from the flu to cancer. They can even be used to boost immunity and prevent disease in some cases. Vaccines, on the other hand, introduce a tiny or inactive part of a live virus to help the body produce antibodies naturally. They can create years or a lifetime of immunity. They cannot help people who are already ill, though. Learn more about antibodies at upmc.com/healthbeat.
So those are the antibodies. Now let's proceed to the antibody structure. So a basic structure of an immunoglobulin molecule resembles the letter Y. It consists of two light identical polypeptide chains located at the top of the Y, and two identical heavy polypeptide chains, two antigen binding sites located at the top of the Y, and an Fc region located at the base of the Y. So the monomer is bivalent in the sense that it has two sites called antigen binding sites. It can bind specifically to the antigen and the antigen determinant that stimulated the production of that antibody. So the acid sequence within the variable regions of heavy and light chains enables the antibody molecule to bind to a particular antigenic determinant. Studies of the gamma globulin component of human blood have revealed that 5 classes or isotypes of immunoglobulins exist, designated the immunoglobulin A, immunoglobulin D, and E, G, and M. So, structures of the different classes of the antibodies. These are the structures. So, um, immunoglobulin G, D, E, and non-secretory A, as we can see, are monomers. As we discussed earlier, monomeres is a Y, resembles the letter Y. So immunoglobulin G, D, E, and non-secretory A are monomers. So secretory immunoglobulin A is a dimer, which is composed of two monomers, as we can see right here. So um, this immunoglobulin M is a pentamer. It composed of 5 monomers. So as we can see right here. So it consists of a J chain. J means for joining. They are short protein chains that bind or that sticks the monomer together. So the secretory piece, which we can see right here, or the component, it protects the immunoglobulin A molecules from proteolytic or protein splitting enzymes and facilitates transport of immunoglobulin A molecules through the membranes. So I have here a table of the immunoglobulin classes. So this one are the classes, and these are the molecular weight. This is the percentage of total immunoglobulin in a serum. This is the approximate. So this right here is the function. So for example, we have immunoglobulin A. It is its molecular weight is 160,000 to 385,000, and it can exist as a monomer or a dimer. So the total percentage of serum is up to 10% to 20%. Its functions is the predominant immunoglobulin class in saliva, tears, seminal fluid, colostrum, breast milk, and mucous secretions of the nose, lungs, and gastrointestinal tract. So we have here an example as well of the immunoglobulin D. Its molecular weight is 180,000 to 184,000. It can be a monomer, so it is less than 1%. So it is found in a large quantities of the surface of B cells. Its function is unknown, but it is possible that immunoglobulin D molecules on the B cell surface serve as antigen receptors and determine which specific antigen that particular B cell is able to respond. So I will not read these tables because it is time consuming. So if you want to look at this, it is in the microbiology book as well as this. So let's proceed to the antibody or antigen-antibody complexes. So it is the combination of an antibody and an antigen. Or AG was what we called AG-AB complex or the immune complex. So antigen-antibody complexes are capable of activating the complement cascade by classical pathway, resulting in, among other effects, and increased phagocytosis as a result of opsonization. So these are the antigen-antibody interactions and their effects. So we have here the interaction. First is the prevention of attachment. So its effects is a pathogen coated with an antibody is prevented from attaching it to a cell. Next, we have clumping of antigen. Its effects is the antibodies linked antigens together, forming a cluster that phagocytes can ingest. Next is the neutralization of toxins. So its effects is the antibodies bind to toxin molecules to prevent them from damaging cells. So these are just some examples of the antigen-antibody interactions in their effects. So if you have time and if you want to look at this, this is also in the microbiology book. So next, how antibodies protect us from pathogens and infectious disease. So here are just some examples on how antibodies protects us. So this is the first examples. For example, a pathogen has entered to a person's body. So for example, it has started to produce a toxin, and that person's immune system responds by producing antibodies against that toxin. So such antibodies are called antitoxin. So the antitoxins recognizes to, bind to, and not neutralize that toxin molecule. So if that happens, they can no longer cause harm. So second example, if viruses can enter the person's body, and we all know that viruses can only bind to host cells that bear the appropriate receptor on their surface. So the molecule on the virus, um, the molecule in the virus that can recognize and binds to the receptor is called the adhesin. So, for example, a person has received a vaccine with an attenuated virus. So when we say attenuated virus, um, it is a vaccine or a virus that doesn't cause harm to a person. So um, so that attenuated virus vaccine stimulates that person's immune system to produce antibodies against that adhesin. So at some later that date, should that same virus enter the person's body, those antibodies will adhere to the adhesin molecules and making it impossible for the virus to bind to host cells. So next example is if a person is infected with a pileated bacterium, that person's immune system responds by producing antibodies against that pili. So that the antibodies bind to the pili, making it possible for the bacterial cell to bind to the pili. So if that bacteria are unable to attach to the tissue, they are unable to cause disease. So next example, or the last example we have here, a person is infected with an encapsulated bacterium. Its immune system, that person's immune system responds by producing antibodies against the capsular polysaccharide molecule. So the antibodies attach to the capsule, and this makes it possible for phagocytes to bind to the encapsulated bacteria. Why? Because that phagocyte have receptors on their surface that can recognize and bind to antibody molecules. So these are just some examples of how antibodies protects us from pathogens and infectious disease. So we have here monoclonal antibodies. So monoclonal antibodies are produced by long-lived cells called hybridomas. So a new long-lived cell or an antibody-producing cell that is capable of producing large amount of cell is called the hybridoma. So the first monoclonal antibodies were produced in 1975. They are usually commonly used in IDPs, or the immunologic procedures used in laboratories to diagnose disease. So with a brief explanation about monoclonal antibodies, is medicines that act like your own antibodies that can help you to stop your symptoms from getting worse and may prevent hospitalization due to worsening of example symptoms of COVID-19. This is not a replacement for the vaccine, but rather it is given to treat your current COVID-19 infection. So next is we have the cell-mediated immunity. Um, wait, cell-mediated immune response involve many types of cells and cytokines. Antibodies are rarely, if ever, involved. A complex of system interactions among many types of cells and cellular secretion are cytokines. So, um, we have a video that is presented last meeting by my fellow reporter regarding this topic, which is the cell-mediated immunity. So I'm just gonna talk about the steps of the cell immunity. So cell-mediated cytotoxic response would include these following steps. First is the macrophage engulfs and partially digests a pathogen, and the fragment is displayed or at the surface of the macrophage. So Th cell binds to one of the antigenic determinants, and Th cell produces cytokines which reach an effector cell of the immune system. The effector cell binds to the target cell, and fourth, vesicular contents of the effector cell is discharged. Last step is toxins produced by the effector cell enter the target cell, causing disruption of DNA and organelles. The target is cell. So here is the video. We have seen this last report of our last report. So we are gonna talk about the natural killer cell. So NK cell, or the natural killer cell, is an subpopulation of lymphocytes called large granular lymphocytes. These cells kill target cells, including foreign cells, host cells infected with viruses or bacteria, and tumor cells. So NK cells lack typical T or B cell surfaces markers. They also differ from T and B cells in other ways. So NK cell activity is not dependent on antibodies. So rather, it has a receptor on the surface for the Fc region of immunoglobulin G antibodies. So we are going to talk about the hypersensitivity, and will be discussed by Mr. Aaron Mina.